Hello friends, welcome to Pharmatronics Online Academy. In this course, we will see about the bioequivalence bioavailability study. The bioequivalence studies are very important for the development of pharmaceutical preparation in the pharmaceutical industry. As you know very well about the generic and branded products. For the approval of generic products as by comparing with the branded product or reference product, the generic product can be approved for marketing authorization by regulatory authorities. So for that, their rationale is the monitoring of pharmacokinetic and pharmacodynamic parameters after the administration of the generic product. There will be a procedure or for the marketing authorization or the permission from regulatory authority, the bioequivalence study data have to be submitted for approval process. So in this course, essentially, this bioequivalence is to ensure uniformity in standards of quality, efficacy, and safety of pharmaceutical products. So, the main purpose of bioequivalence study is to maintain the quality, efficacy, and safety of pharmaceutical products. Reasonable assurance is to be provided that various products containing same active ingredient marketed by different licenses are clinically equivalent and interchangeable. As I have already told that for permission or the approval of generic product for the marketing purpose, it should be bioequivalent or therapeutic equivalent. So for that, the clinically equivalent and interchangeable data have to be submitted. Release of an active substance should be known and reproducible. The release of active substance means the dissolution in case of tablets and capsules should be known and reproducible. Both bioavailability and bioequivalence focus on release of drug substance from its dosage form and subsequent absorption in circulation. Means if you are administering or patient administered the oral dosage form, that is the tablet, the bioavailability and bioequivalence is determined by its release of a drug substance from the tablet and subsequently it gets absorbed into the circulation. The plasma concentration of the drug substance is measured using analytical methods. Similar approaches to measure bioavailability should be followed in demonstrating bioequivalence. For demonstration of bioequivalence study, there should be a procedure, a protocol, synopsis. A specific procedure needs some guidelines. As per the guidelines, the bioequivalence study have to be performed. So let's see, in this course, you will learn about the bioequivalence and bioavailability study. So step by step, session by session, we will see how this bioavailability and bioequivalence study is performed in patients and how to get bioequivalent product or how to submit the data. Okay, let's see. First, bioavailability. It means the measurement of relative amount and rate at which the drug from administered dosage form reaches the systemic circulation and becomes available at site of action. As we have told about the after administration of oral dosage form, after administration it reaches the systemic circulation after absorption, but before absorption it should be dissolved in the intestinal fluid or any body fluid so that it comes into the systemic circulation through some channels, so through some permeable membranes and so on, and becomes available at the site of action. So how will you calculate bioavailability, that is the bioavailable fraction? It refers to the fraction of administered dose that enters the systemic circulation. Means the after administration of 100% of dosage form, means 100% drug is administered, but it cannot be bioavailable, means it is not available at the site of action as 100% or out of systemic circulation. So here is a fraction of bioavailable dose, means bioavailable dose divided by administered dose, which gives the fraction of bioavailability. The therapeutic relevance. Here a graphical representation of therapeutic relevance. Let's see one by one. A plasma drug concentration versus time, a graph representation which represents a bell-shaped graph which shows the administration of a drug from this point comes up to this point, there will be a onset of action and after that absorption phase it reaches to the maximum concentration. After that there decreasing portion shows the post absorption phase and elimination phase. It means the concentration of that drug substance is eliminated or decreasing. So the duration of action starts from this point, and up to this point the onset of action is maintained. Here the therapeutic range, the space between the minimum effective concentration and maximum safe concentration, that is called therapeutic range. Between this range the safe effective concentration is maintained. Beyond this limit there is a toxic level, means beyond this absorption level there will be a toxic effects will be observed. So beyond the maximum safe concentration there is a toxic level, and below the therapeutic range subtherapeutic level, means no therapeutic action is shown by that dosage form. Then absolute bioavailability. It compares the bioavailability of active drug in systemic circulation following non-intravenous administration with the same drug following intravenous administration. Means we have to compare dosage form which are administered through oral and one is through intravenous administration, but both the dosage form containing same amount of drugs. For drugs administered intravenously the bioavailability is 100%, and we have to determine the best administration route, which administration route is preferable for that drug substance alternative to the intravenous administration. Absolute bioavailability is measured using the ratio of area under curve of the drug divided by area under curve, means the IV route, the drug absorption or bioavailability by IV route. The absolute bioavailability of nimodipine for different routes. Here one example is given. A graphical plot represents the plasma concentration versus time in minute. Here three route of administrations are selected, that is oral, nasal and IV. The oral route of administration shows the bioavailability is negligible, like 1.17%, and nasal bioavailability is observed that 67.4%, and intravenous bioavailability is 100%, this one. So the graphical representation shows the preferable route of administration and preferable route of administration. Relative bioavailability. It compares the bioavailability of a formulation A of a certain drug when compared with another formulation B of the same drug, usually an established standard. Means if any formulation in different dosage form we have to compare with another dosage form with the same drug, so we have to find out relative bioavailability between these two dosage forms. Means formulation A is tablet and formulation B is IV route or any parenteral formulation. So we can relatively find out the relative bioavailability between these two formulations by using this formula: area under curve for the drug and area under curve for a standard. Means test product and reference product are calculated. Then plasma concentration versus time profile. Here a graphical representation for two formulations, formulation A and formulation B, shows the different behavior for absorption. Here the formulation A starts with this onset of action and then peak maximum concentration, that is the Cmax. Here the elimination, and formulation B shows this type of graph. The Cmax is here and the elimination. You can see the difference between two absorption levels and plasma concentration versus time, means how the absorption, at which rate the two formulation shows absorption, and the therapeutic range for these two formulations is shown here. Then how will you calculate the area under curve? By using trapezoidal rule. Any graphical representation shows a bell-shaped graph from initial zero level to the some amount of drug concentration is up to the maximum concentration, that is Cmax. Then after release, the concentration becomes reduced and the elimination starts. Up to the elimination, the graphical representation is plotted. After the plotting, some points are collected, sample points at different time points. So these time points are joined with the time period, so a trapezoid is formed. Using this trapezoidal rule, you can calculate the amount of or area under curve, means concentration in that area. So this formula can be used for determination of area under curve. Area under curve between these 2 points 2 to 3 means concentration for this second point and concentration for this third point, mentioned as Cp2 and Cp3. The formula indicates the Cp2 plus Cp3 divided by 2 multiplied by T3 minus T2. That is the time required for these respective concentration. Hope you have understood these all the things as we have seen in this session. So in detail we will see in the next coming sections. So thank you.